Welcome back to BioBonanza. If you couldn't tell by the intro, today we're talking about cells. Let's begin. One of the classifications for cells is whether they're prokaryotic or eukaryotic. When compared to each other, prokaryotic cells are smaller and less complex, and eukaryotic cells are larger and more complex. Eukaryotic cells are more complex because of things inside of them called organelles. Organelles are membrane-bound structures that have a specific function. Despite these differences, they do have some things in common. Both prokaryotes and eukaryotes have a plasma or cell membrane, ribosomes, DNA, and cytoplasm. The cytoplasm is the area inside of a cell, and it includes everything that is found in it. Structures in the cytoplasm are suspended in a jelly-like fluid called cytosol.
Artlist.io.
In prokaryotes, DNA is a single circular chromosome and is contained in an area called the nucleoid. And in eukaryotes, there are multiple linear chromosomes that are located inside of the nucleus. Ribosomes are protein-making structures found in the cytoplasm that are created when a small and large subunit combine. They read a copy of DNA called mRNA and make a protein depending on its code during a process called translation. Lastly, there's the plasma or cell membrane. The plasma membrane is made up of phospholipids, and since they have a head that is attracted to water and a tail that is not attracted to water, they form a bilayer where the heads are facing outwards and the tails are facing inwards. In addition to this, there are proteins that are embedded in the membrane. Some are integral, which means they're always embedded in the membrane, and others are peripheral, which means they're attached to the membrane temporarily or to other proteins temporarily.
Music license.
Now, let's move on to what makes eukaryotes special, their organelles. But there is one more classification for eukaryotes. This classification is whether it is a plant, animal, fungi, or a protist. Just like prokaryotes and eukaryotes, these cells all have their similarities and differences. Let's go through the organelles that they all have in common. First, the nucleus. The nucleus is surrounded by a membrane called the nuclear envelope, which is a double-layer membrane. There are also multiple nuclear pores in this membrane that are surrounded by guard proteins that only allow certain things in and out of the organelle. Within the nucleus is DNA, and there is also an area inside called the nucleolus, which is where ribosomes are made. It's the darker spot. Next, there's the endoplasmic reticulum, but I'm just going to say the ER for short. The ER is a mass of membrane folds that are cut off from the cytoplasm. This gives the inside of the organelle a unique environment. There are two types of ER, the smooth one and the rough one. The smooth ER creates lipids and detoxifies the cell, and the rough ER, which is studded with ribosomes, giving it the rough appearance, produces and processes specific proteins and eventually sends them off to be further processed. Vesicles are membrane-bound pouches that are moved from place to place within the cell. They can hold a multitude of things inside of them. For example, they carry lipids that are created by the smooth ER towards the plasma membrane so they can be secreted. Fourth, the Golgi apparatus. The Golgi apparatus is similar to the ER since it has a unique internal environment. When proteins are dropped off to the cis phase, they move pouch to pouch where they are further modified and given a molecular ID tag that determines where they go. Eventually, they make it to the trans phase and are packaged in a vesicle and sent to their destination. The mitochondria, which is plural, or mitochondrion, which is singular, is a special organelle due to the fact that it has its own DNA. This DNA is similar to prokaryotic DNA, and it also has its own ribosomes and tRNA. Lastly, it grows and replicates in a way similar to how prokaryotics do. The main function of the mitochondrion is to use sugars to create energy for the cell using a process known as cellular respiration. Next, lysosomes. A lysosome is similar to a vesicle, except inside it has hydrolytic enzymes, and its internal pH is about 5, making it acidic. These hydrolytic enzymes allow it to fuse with other structures to break them down or to facilitate indigestion. Peroxisomes. Peroxisomes are also membrane-bound pouches. They help with lipid metabolism and detoxification. They also sometimes contain a crystalline core that has many enzymes inside of it that helps it to perform these tasks. Finally, the cytoskeleton. The cytoskeleton is a network of proteins that help to do many things, like determine the shape of the cell, aiding in its shape changes, anchoring organelles, and moving certain structures like chromosomes or vesicles. The main fibers from the smallest to largest are microfilaments, intermediate filaments, and then microtubules. These proteins also make structures that are used for movement located outside the cell. These things are cilia and flagella. Flagella move from side to side, and cilia move using a sweeping motion. Here's an example of a motor protein moving cargo along the cytoskeleton. Now, let's see what differences they have. First, the extracellular matrix. The extracellular matrix is found outside of animal cells, and it is a network of protein fibers, mainly collagen. It helps to anchor proteins that are in the plasma membrane, connect neighboring cells, and also relay signals to other similar surrounding cells. Vacuoles are membrane-bound pouches that are able to store many things like food, water, ions, and pigments. The vacuoles of animal cells are significantly smaller than the vacuoles of fungi and plant cells, but the vacuoles for protists can vary in size. Also, plants, some fungi, and some protists have a central vacuole that only stores water and ions. The fluid inside of this structure is called cell sap. There are also other types of vacuoles, such as contractile vacuoles that store water and then squeeze it out later on. These are commonly found in protists. Third, chloroplasts. Chloroplasts are organelles that are found mainly in plant cells, but they can be found in some protists. Just like the mitochondrion, chloroplasts have their own DNA that is similar to prokaryotic DNA, their own ribosomes and tRNA, and they grow and replicate in a way similar to how prokaryotes do. Their main role is to use energy to create sugar for the cell through a process called photosynthesis. The cell wall. The cell wall can be found in plants, fungi, and in some protists. The cell wall is the outermost structure that protects, supports, and reinforces the cell's structures. In plants, the cell wall is mainly made up of cellulose, and in fungi, it is mainly made up of chitin. In protists, its composition can vary. Some other structures that I didn't mention are pseudopodia and extrusive organelles. These structures are found only in protists. Protists use pseudopodia, which means false foot to move, and extrusive organelles are organelles that can be discharged from the cell in response to something. An example are trichocytes, which are dart-like structures used for defense or capturing prey. Thanks for watching. Like and subscribe for more videos just like this one.